Very good evening, everyone. Please confirm if you all can see the screen. I hope the screen is visible to you all. Please let me know in the chat. So welcome to another session of Brahmastra Series. And today we will be revising and learning the concepts from subunit C of unit 13, that is Biophysical Methods. Okay, thank you for confirming, Sarita, and very good evening. So how's your preparation going on, everybody? I hope you all are revising well, practicing questions. So this 1 hour, we will be revising and practicing the questions from the topic biophysical method. So I hope you all know biophysical method. Subunit of unit 13 is all about spectroscopy, right? Where you study about UV visible spectroscopy, or you study about fluorescence spectroscopy, you study about circular dichroism, NMR spectroscopy, mass spectrometry, right? And often in CSIR NET Life Science exam, you will find questions on spectroscopy techniques, either part B or part C. So question can be either very direct, part B question, memory-based question on the application of the technique, right? And part C question, it can be experimental-based question where they can give you a spectrum, a figure-based question, and some statement based on that, and you have to answer the question going through the figure given and the statements given. So let's get started. Let's begin with the very first question. So I will be giving you some time to solve this question. So question is, protein A was subjected to different experiments. First, SDS page with and without beta mercaptoethanol. Then second is fluorescence spectra. Third is far UV, and fourth is near UV circular dichroism spectra at pH 7, indicated with blue line. So this is blue line, and this one is red. So the difference is in the pH, where the blue one is at pH 7 and the 3 at pH 3, the red line is indicated by red line. The spectra at pH 3 is indicated by red line. So results are shown below. Which one of the following options represents the correct interference? First is protein A is an SS bonded homotetramer, and each subunit has a molecular mass of 50 kilodalton, folded at pH 7 and molten globule at pH 3. Second, protein A has a molecular mass of 200 kilodalton. Beta mercaptoethanol degrades the protein. Low pH changes the conformation from alpha helix to beta sheet. Third statement, SDS denatures protein A into different oligomeric states. Low pH changes the conformation from alpha helix to beta sheet. And fourth is SDS promotes the formation of different oligomeric states of protein A. Low pH changes the conformation from beta sheet to alpha helix. So solve this question and let me know what is the answer here. So strategy of solving this question is that you just have to go through the figure given, okay, the experimental data which is given, the one we are getting the result of SDS page, the fluorescence spectra, far UV, near UV circular dichroism spectra. Also, you need to be familiar with the terms like what is homotetramer to answer this question, right? What is the role of beta mercaptoethanol? What is the role of SDS, right? What is SDS, right? And then far UV circular dichroism or near UV circular dichroism spectra. So, which region of UV visible, which region of ultraviolet radiation is used to determine the secondary structure, tertiary structure? Accordingly, you can answer this question. So you don't have to think out of the box. Okay, you don't have to think out of the box. You just have to look at the experimental results which have been given. And based on that, you have to find out that which of the following options provide the correct inference. Okay, Sarita, you are saying option 1. Okay, good. Okay, Rinku, option 1. Very good. So, really appreciate you both for trying. I hope others are also practicing. Yes, everyone has to solve and let me know the answers in the chat. These type of questions are very scoring and you can often get it in the exam. So as I told you, to solve this question, you should be familiar with the role of SDS, beta mercaptoethanol, what does far UV, near UV circular dichroism spectra indicates, right? So always remember, in order to determine the secondary structure, right? So secondary structure, you have alpha, helix, beta sheet structure, right? And tertiary structure is your folded structure, right? Three-dimensional structure. So basically, always remember, the circular dichroism spectroscopy is very popularly being used to study the secondary structure of proteins. And mainly the far UV region, okay, is being used to determine it is used to determine the information regarding the kind and quantity of secondary structure. And near UV spectra and tryptophan emission both have been used to determine the tertiary structure. Okay, so tertiary structures, they have been revealed by tryptophan emission and near UV spectra. Now let's go in detail and understand how these spectra can help us in determining secondary or tertiary structures, or how tryptophan emission can help in determining the structures. So first of all, as you can see, like we'll go through each figure one by one, each experimental result one by one. So here first is of SDS page. So SDS is an anionic detergent, right? SDS is an anionic detergent. And basically in polyacrylamide gel electrophoresis, we add this SDS because proteins based on their amino acid residue, like amino acids are building blocks of protein. So based on the side chain, amino acids can have a positive or negative charge, right? So SDS, it linearizes the protein. That is, it breaks the non-covalent interaction in a protein and convert it into a linear form and provide a uniform negative charge to the protein. So this is why the proteins now, because they have a net negative charge, they can migrate from negative to positive electrode when electrophoresis is performed. Now, along in the SDS page, we are adding beta mercaptoethanol, right? We are treating the protein with the beta mercaptoethanol. So you can see in presence of beta mercaptoethanol, we are getting a single band of 50 kilodalton. However, when beta mercaptoethanol was not added, then we are getting a single band of 200 kilodalton. So that means, like, what is the role of beta mercaptoethanol? It breaks the disulfide linkages, right? So the point is, that means the molecular mass of a protein is 200 kilodalton, but when you treat it with beta mercaptoethanol, the disulfide linkages are being broken, and that means the protein might be comprising of several subunits. Because 50 kilodaltons indicates what? 50 into 4 gives you 200. So that means 4 subunits are there, and that too, they are all not different, they are same, right? Because we are getting a single band. So that is only possible when all the 4 subunits are same. So when a protein is subjected to SDS-gel electrophoresis and presence of beta mercaptoethanol, I told you it shows a band of 200 kilodalton. However, in presence of beta mercaptoethanol, you are getting 50 kilodalton, right? Which reduces the disulfide bond, leading to separation of protein subunits. And we are getting and 50 kilodalton means like total 4 subunits might be there. So it will be a tetramer. And because it's a single band we are getting, so it should be homotetramer, right? Now, so this information we can conclude by seeing this SDS page result. Second is fluorescence spectra. So you can see that in fluorescence spectra at pH 7, we are getting the fluorescence intensity. Like you can see 330 nm, like when you are exciting the protein at 290 nm. So fluorescence is when the light emitted is of longer wavelength than the light absorbed. So 330 nm, and at pH 3, 350 nm. So there is an increase, right? And if you see far UV circular dichroism spectra, so as I told you, far UV circular dichroism spectra shows the type and quantity of secondary structure remain unchanged. So here you can see there is no considerable change here, right? Like if you see the result, both the red one and the blue one, there is no considerable change. But in near UV, you can see that this red line becomes flat. This red one becomes flat, right? So if we go through each statement, each option one by one, so option one is that protein A is an SS bonded homotetramer. So yes, this I have just now told you that yes, it comprises, it's a 4 subunits and they are all same so homotetramer and they are connected with a disulfide bond. This is the reason that without beta mercaptoethanol, we are getting a single band of 200 kilodalton. But on adding the beta mercaptoethanol, we are getting a single band of 50 kilodalton. So that means each subunit, they have a molecular mass of 50 kilodalton. Now, it is also mentioned folded at pH 7 and molten globule at pH 3. Now, what is molten globule? Molten globule refers to protein state that are more or less compact. That is why we use a term globule. Okay. But they are lacking the tight packing of amino acid residues, which creates solid state like tertiary structure of completely folded protein. So hence, we are using the term molten. Okay, or in a very simple way, you can remember it's a compact intermediate in which tertiary structure of protein is lost, but secondary structure is intact or even strengthened, right? So in the beginning, I told you far UV, it tells us about the secondary structure. So it is unchanged. But in near UV, you can see the red becomes completely flat. So it indicates loss of tertiary structure, right? So yes, it can be molten globule at pH 3. So this confirms our result, and that means answer can be option 1, right? Then if you go through, let's check other statements also. So if you see option 2, it states that protein A has a molecular mass of 200 kilodalton. Beta mercaptoethanol degrades the protein. No, it doesn't degrades the protein, right? It basically, as I told you, it reduces the disulfide bond. It's a reducing agent leading to separation of protein monomer. Then third, so this cannot be correct. Third is SDS denatures protein A into different oligomeric state. Low pH changes the conformation from alpha helix to beta sheet. So as the far UV tells us about the secondary structure, we don't see any sort of considerable change over here, right? And statement 4 is SDS promotes the formation of different oligomeric states of protein A. Low pH changes the conformation from beta sheet to alpha. So again, this is incorrect because as I told you, far UV, we are not seeing any considerable change. And if you see the fluorescence spectra, like what does this increase in the wavelength indicates? That tryptophan residues must have been exposed as a result of denaturant treatment, right? Like when you added the SDS, so when the folded protein it linearizes, it exposes the, like its tertiary structure is lost, it exposes the tryptophan residues, as a result of which there is an increase in this from 330 to 350, right? So this further confirms our result that yes, answer here is option 1. So Rinku and Sarita, well done, you have answered it correctly. So to learn concepts like these, to learn the strategies of solving the questions, especially part C, CSIR NET Life Science questions, you can always attend this Brahmastra series. But if you want to prepare for CSIR NET or GATE exam, then we have a Lakshya batch, upcoming batch, so you can go through it. So for details, you can call on our toll-free number, and even you can visit to our store.biotecnika.org. Also, those who don't know, we have launched the T3 test series, which comprises of 10 toughest questions, unit-wise questions, part C questions. So since now the time for the exam left is very, like, since now you have only few days left for the exam, right? So you can utilize these days, download the campus app, and you can Biotecnika campus app, and you can get access to this T3 test series, and you can practice every day 10 toughest questions, and that to unit wise part C questions. So to get access of this, you can again call on our toll-free number. So please tell your friends and others who are also giving this exam, CSIR NET Life Science exam, to get access to this T3 test series. And if they want to join the classes, regular batches, like if they want to prepare for the CSIR NET or GATE exam, then they can join our Laksh batch. Now let's discuss another question. So again, here you can see it's a part C question, a figure-based question. Absorption spectra of L-tyrosine in acidic, represented with the continuous line, that is this one, and basic, that is with the dotted line medium, was estimated and plotted on a graph as depicted below. Following interpretations were made. A. Change in pH from acidic to basic results in shift in lowest energy absorption maximum and decrease in molar absorptivity. B. Shifting of absorption band to longer wavelength signifies a shift to lower energy, also known as redshift. C. Shifting of the absorption band to shorter wavelength signifies a shift to higher energy, also known as blue shift. And wavelength shift is always accompanied by change in intensity of the absorption band. Select the combination with correct interpretations. So like a graph is given and some interpretations are given, and based on this graph, you have to determine like like you have to select the statements or you have to select the correct interpretations. So again, you don't have to think out of the box. Just go through this given graph carefully. But definitely, you need to be familiar with the terms like what is redshift, what is blue shift, what happens when the absorption band shift to longer wavelength. So, whether the energy increases or decreases, or when the absorption band shifts to shorter wavelength, so energy increases or decreases. So, these basic concepts should be clear to you. Then only you can answer this question. So, again, this question is not very difficult if you are familiar with these terms and if you know how to read this graph, you can easily answer this question. So please solve everyone. So I want everyone to try and please let me know the answer in the chat. I hope you all are solving. Even you can omit options 2 out of 4. Like if you say statement A is correct, then definitely third and fourth cannot be the answer. Or like if you say D is correct, so obviously first, second, and third. Like if D is correct, then first, second, third cannot be the answer, right? So this way also you can like use this strategy, omit 2 options out of 4 if you are getting confused between any 2 statements. Okay, Sarita, you are saying option 3, and Rinku, you are saying B and C. That is option 3 only. Okay. So let's check. So first of all, as I told you, you need to be familiar with the concept of what is redshift and what is blue shift. So I want you all to have a look at this figure. So we use this terminology for shift in absorption spectra. So always remember, so on x-axis you can see we have a wavelength, and on y-axis we have a molar absorption coefficient. So always, whether it's a substituent or whether the solvent, okay, like whether a protein shifted to polar environment or non-polar environment, or if there is any group attached, so they all have the effect on the absorption spectra. So bathochromic shift or redshift term is used when the shift of absorption maxima to longer wavelength. What is absorption maxima? It is the wavelength at which maximum absorption of light occurs, right? So basically, let's say the protein absorbing around, let's say your sample is absorbing around 280. Now there is an increase in, like, so this is, like, you can see on x-axis we have wavelengths. So the wavelengths are written. So like 240, 250, 260. Just a hypothetical example I'm giving you. Okay, 280, 300, right? And so on, right? Now you, and on y-axis molar absorption coefficient, right? So initially, let's say a protein absorbs at 280, but because of change in the environment, either the absorption can increase or decrease. So let's say it increases to like 300, right? So whenever, so what happens here, the lambda max moves, increases from 280 to 300, right? Or like similarly, let's say, simple example, let's say you have a benzene. Now to this benzene it shows a lambda max at a particular wavelength, right? But if to this benzene you attach a OH group, phenol, or if you attach a NH2 group, aniline, so the lambda max will be different for for this phenol and aniline in comparison to that of benzene. Okay, so when there is a shift of absorption maxima to longer wavelength, we use the term redshift or bathochromic shift. And see, you don't have to do rote, like simply memorize, you can understand. We know that white light has 7 colors, right? Vibgyor: violet, indigo, blue, green, yellow, orange, red. So red lies towards longer wavelength, right? In general, if you write down the range of visible light, it is 400 to 700 nm, right? This is what in round off we write. So red region lies towards the longer wavelength, 700, and blue region lies towards the shorter wavelength, right? So R is red shift, so that means towards longer wavelength. On the other hand, hypsochromic refers to, also known as blue shift, where the shift of absorption maxima to shorter wavelength. That is, let's say you have 280, but now it shifts from 280 to like around 250 or like that. So it is blue shift. Lambda max shifted to shorter wavelength. Similarly, if the intensity, absorption intensity of a compound increase, okay, so like this is your molar absorption intensity. Now, if it increases, then you have a hyperchromic effect, and if it decreases, it is hypochromic effect. So these terms you have to remember to answer the questions. Now, since now we are familiar with these terms, we will now go through the interpretations given in the question one by one. Before that, you can see this is the graph given to you. So in the graph, wavelength is on x-axis and y-axis shows the absorbance. So here you can see this is the continuous line and this is the dotted line. So it is just the difference, like acidic and basic medium, it is showing, readings it is showing. So interpretation is change in pH from acidic to basic results in shift in the lowest energy absorption maxima and decrease in molar absorptivity. See, by looking at this graph, we know that certainly if you compare the wavelength range of this in the acidic medium, the absorption in acidic medium and basic medium, so definitely basic medium is towards the longer wavelength, right? But as such, there is no information indicating the specific position of lowest energy absorption maximum, right? So that is why this statement is incorrect. Because it states what? Shift in lowest energy absorption maximum and decrease in molar absorptivity. So here we are not, like, it is not been specified, does not specify any shift in lowest energy absorption maxima. No information, specific position of lowest energy absorption maxima is not given. That is why it is incorrect. So any option having a statement, a interpretation cannot be the answer. So in the options 1 and 2, A was given. So that means 1 and 2, like you can, out of 4 options, 1 and 2 cannot be the answer. Now you're left with 3 and 4. So B is shifting the absorption band to longer wavelength signifies a shift to lower energy, also known as red shift. So just now I told you that red shift is bathochromic shift, right? So if there is a shift absorption band to longer wavelength, certainly it's a red shift. And you all should know that the relation between wavelength and energy is inversely proportional. Okay, higher the energy, longer the wavelength. So HHS you can remember, or shorter the, higher the energy, shorter the wavelength. Okay, please correct this. So HHS, higher the energy, higher the frequency. So energy and frequency are directly proportional, wavelength is inversely proportional. Right? So when I say longer wavelength, that means lower energy. So even you can learn it this way also, 3L. Okay, these keywords you can remember. Lower the energy, lower the frequency, longer the wavelength. Okay, this is indicated by 3L. So statement B is absolutely correct, because just now, like few minutes back, I told you that here, if you see the dotted line, which indicates basic medium, absorption band in basic medium, they appears at longer wavelength compared to acidic, like you have to check the wavelength. Okay. So the absorption band in the basic medium appears at the longer wavelength compared to the acidic medium, confirming a redshift. Then statement C, shifting of absorption band to shorter wavelength signifies a shift to higher energy, also known as blue shift. So higher energy and shorter wavelength. This is also correct, right? This is also correct. And D is wavelength shift is always accompanied by change in intensity of absorption band. No, that is not always the case. Sometimes it can be accompanied by change in intensity of absorption band, but it is not always the case. Okay, so B and C is given in option 3, and therefore it is the answer. Option 1 or statement interpretation 1 you are saying, right? So that is what I told you, Sarita, here, that this statement is incorrect. If you're asking about statement A, then this is incorrect, because it states that lowest energy absorption maxima shift upon changing the pH from acidic to basic, right? But however, the given information, whatever information given to us, does not specify any shift in lowest energy absorption maximum. So that is why the graph provided only shows the absorption spectra in acidic and basic medium without indicating specific position of the lowest energy absorption maximum. And in general, you are asking what is the relation of absorption with wavelength. In general, absorption depends upon, like when we talk about maximum absorption, it varies from a sample to sample, right? Like for protein, the maximum absorption occurs at 280 nm. For DNA, it occurs at 260 nm. Now, the reason being the answer lies in their structure, right? Like if you see the DNA, then nucleic acid, then mainly it's the nitrogenous bases responsible for absorption of light. For proteins, it is the aromatic amino acid. But it's not that, okay? All solvent substitution also plays a very important role. Okay, so concentration absorption, if you are asking, so yes, in general, absorption is directly proportional to concentration. Beer-Lambert's law, if you remember. More the absorbance means more the concentration, or more the concentration means more the absorbance, right? Like if you have just a simple example, if you have an empty cuvette and if you pass the light through it, nothing will be absorbed, right? All the 100% of light will be transmitted, right? So transmittance 100%, absorbance 0. But if I fill in something inside it, let's say DNA, so what will happen? This is a, like when you have a matter, it will absorb the light. So what will happen when you pass the light? The DNA will absorb the light, and absorption is directly proportional to concentration of the sample. More the DNA, more the light will be absorbed. I hope this answers your question. Is this what you are asking, right? So directly proportional relationship. Absorbance is directly proportional to concentration and even path length. Got it? Okay, that's great. Coming to next question. Shown below are the circular dichroism spectra of a protein recorded under two different conditions, right? A and B. So from the options given below, select the one that is best interpretation of the spectra. First is protein has a helical secondary structure under condition A, that is denatured under condition B. Second, protein has a helical secondary structure under condition A, that is converted to beta sheet under condition B. Third is the spectra represents tertiary fold of the protein with condition A corresponding to mixed alpha helical plus beta sheet fold, and condition B corresponding to largely beta sheet fold. And fourth is difference between the spectra under conditions A and B is due to lower protein concentration under condition B. Now, this question is based on circular dichroism spectroscopy. And if you have studied about like at what, like if you have studied about how to read the circular dichroism spectra, then only you can answer this question. You should be familiar with what positive and negative bands are indicates presence of alpha helix structure or beta sheet structure or random coil structure, right? Now, Sarita, that is, you are asking A is alpha helical and beta is random coil. Now, see, if I tell you this, you will get to know the answer only. That is what I'm saying. You need, by seeing the spectra, you should understand, like by looking at the positive and negative bands, you should make out like whether it's a spectra of alpha helix or beta sheet. Okay, so you just try, because if I tell you right now, you will just know the answer only. Okay, Sarita, you have answered option one. What about others, Rinku? Please try, everyone. At the end in the exam, you only have to solve the questions. I will not be there to help you out, right? So at least just think of like what strategy you will apply here, which concept you will apply here in solving this question. And please remember, everyone, those who will be watching the session, 1 hour session, like those who will be attending live this session, will be getting practice questions in the end as a freebie along with this Brahma series session. So stay tuned, stay uh, and practice questions during this live class. And at the end, you also will be getting some practice questions. The link for it will be shared at the end only. But it will be for those only who will be attending this live session. Okay, Rinku, you are saying option 2. Fine. So let's check. So as I told you few minutes back that to answer this question, you need to be familiar with the circular dichroism spectroscopy and how to read the circular dichroism spectra. And also you need to be familiar with the wavelength at which the like positive bands, negative bands are being seen at a particular wavelength for alpha helix structure, beta sheet, and random coil. That is unordered conformation. So always remember far UV circular dichroism, it provides information about the kind and content of secondary structure. This I have already told you when we were doing the first question. So always remember, how do you know that alpha helix structure is present in your protein, right? So basically, always remember, if there is a alpha helix structure, you will get two negative absorption bands centered around 208 nm and 222 nm. And this is mainly because of the, like, depending on the type of transitions happening. So if pi to pi antibonding transition happening, then the absorption band is observed at 208, and that to negative band, and to add another at 222 nm, that is because of non-bonding to pi antibonding transition. So this concept of transition I have already covered in detail in our regular classes, right? And strong positive band around 192 nm because of pi to pi antibonding transition. So here in this particular spectra given, you can see wavelength is given on x-axis and ellipticity is given on y-axis. So we can measure the circular dichroism spectra can be plotted ellipticity versus wavelength. So here you can see beyond 0, it indicates the positive bands, and below 0, it indicates the negative bands. So as I told you, two negative bands. So you can see this is one negative band. Okay, this one will be the negative band. And this is the another negative band. So one is around 208 and another is around 222 nm. So presence of these two bands indicate alpha helix structure, and positive band around 192 nm. Okay, so you have to learn it. Then how will you know that whether beta sheet is present or not? So for beta sheet, you have to see the blue line. Beta sheet is always characterized by a negative band centered around 216 to 218. So if you see this, this is the negative band for the beta sheet and centered around 216 to 218. And a positive band is centered around 195. Okay, so this is 195 nm. Then random coils can be determined. Okay, if you see the random coil, so you can see this is the negative band. You can see it is somewhat over here, and a weak positive band is centered over here, right? So in this question, actually, so the point is you have to learn the wavelength, okay? What all bands are absorbed at what wavelength for alpha helix and beta sheet and random coil, okay? So as in this question, if you go through the given spectra, you can see this is for A and this is for B, right? Now, Sarita, you asked me that, ma'am, whether A is alpha helical. So how can we check? We can check like if we are getting a positive band around 192. So this is 190. So yes, this is around 192, right? And negative band, if you see, right? So negative band is this one is this one, another is this one. So one is around 208 and you have another one which is around 222. So yes, this confirms that A is alpha helix, right? So protein in A condition shows a strong positive band around 192 and two negative absorption bands centered around 208 and 222, indicating presence of alpha helical secondary structure. And for beta, if you see, so beta is represented with this B1, that is the black dotted line. So for beta condition, you can see that it is coming like it is becoming flat over here, right? Ideally for beta, you should get like this result, right? Positive band and the negative band, but here it is becoming flat. So if we go through the statements given, the protein A has a helical secondary structure under condition A. So absolutely right. Alpha helix is secondary structure, alpha helical secondary structure. But you can see that it is denatured under condition B, right? So in condition B, the line becomes flat, right? So this is not, you cannot say that B is beta. Okay, actually this is showing you the condition. A is definitely for alpha helix, but B is the condition where you can see the structure, this portion is becoming flat. So A and B are the conditions, right? Where A indicates, A condition definitely indicate presence of alpha helical. B is not indicating beta sheet, but it is indicating that line become flat, indicating complete loss of secondary structure, which is possible only when protein is denatured under condition B. So this confirms our answer, and therefore answer is option 1 here. Okay, so this part, if you see statement 2, if you go through other statements, it says that the protein has a helical secondary structure under condition A. Okay, so this is common to the first statement, but this part is different, that is converted to beta sheet. If it would have been converted to beta sheet, then we would have got the right 195 positive band, 190 positive band at 195 nm and all, but we are not getting anything here like that. And third is that spectra represents tertiary fold of the protein with condition A. This is incorrect only because when I say alpha helix, it's a secondary structure. And fourth is difference between spectra under condition A and B is due to lower protein concentration under condition B. Now, no information about concentration is given over here. And moreover, as we are talking about the circular dichroism spectra, based on the information given, it is about the positive bands and negative bands we have to look at. And that is why the most appropriate statement here is 1. So answer here is option 1. So, Sarita, you have answered correctly. Very good. Then moving over to another question. From the following statements, choose the option with all correct statements. So, statements given are A. Surface plasmon resonance can be used to determine binding constants only on range of 10 raised to power 2 to 10 raised to power 3. De novo sequencing is not possible by mass spectral methods. C. Position of hydrogen atom in protein is not directly determined by X-ray diffraction. And D. Circular dichroism and NMR, that is nuclear magnetic resonance spectroscopy, do not give the same information on protein structure. You have to choose the option with all correct statements. Now, kindly note that this is a statement-based question. So far, we have discussed about the figure-based questions, right? The graph-based, the spectrum-based questions. Now, in the exam, part C question can be a statement-based question also on spectroscopy technique. And here you can see that to answer this question, you need to be familiar with mass spectrometry, you need to be familiar with circular dichroism, NMR spectroscopy application, even X-ray diffraction, right? So, when you are preparing subunit C of unit 13, make sure that you learn all the spectroscopy techniques. Okay, you learn all the spectroscopy techniques. It's not that you prepare circular dichroism only or NMR only. So if that would have been the, if you're doing that way, then if such type of question is asked, you will not be able to solve it. Right? So make sure you prepare all. At least be familiar with the principle and application of each spectroscopy techniques. And the best way of learning is compare and learn. So tell me what is the answer here? So Sarita, you asked me this question regarding the previous, related to previous question, right? That A is alpha helical and B is random. So I hope it is clear to you now, the concept. Please let me know in the chat. And why others are not responding? I want response from everyone. So to answer this question, you should know what is de novo sequencing, what is the role of X-ray diffraction technique, and especially this statement, circular dichroism and NMR spectroscopy, whether they give the same information on protein structure. Same information means what? Like whether they both are used to determine secondary structure or tertiary structure. With that context, it has been asked. So this statement has been asked several times in the CSIR NET Life Science exam, where this they can ask you simply like which of the following technique can be used to determine secondary structure of the protein, or alpha helix and beta sheet structure of the protein. And in the options, they give you circular dichroism also and NMR spectroscopy also. So many students, they get CSNET aspirants, they get confused. Like according to them, both can be used. But to be more precise, any one is used for secondary and other is used for tertiary. So that you are going to know today. And some of you might already be knowing about it. So this statement is very important. So when you are studying about spectroscopy, make sure you emphasize on these things. That is why I'm saying compare and learn. Both are absorption, like circular dichroism is absorption spectroscopy technique. In NMR, also you like the only difference is in circular dichroism, we take help of UV rays. In NMR, we use radio waves and magnetic field. But you can determine the structure of proteins using both. But to be more specific, which structure, secondary or tertiary? Okay, so Rinku, you are saying option A, B, and C. So 4 statements when are given to you, at least whichever 2 you are most comfortable with or familiar with, you can answer that question. Based on that, you can answer this question. Okay, let's say you know about mass spectroscopy. So if this is correct or incorrect, based on that, you can omit options because you have to choose the correct statement. Option with all correct statement. So let's go through each statement one by one. Surface plasmon resonance can be used to determine binding constant only on this range. So no, this is incorrect. From micromolar to nanomolar range, it can be used. So that is why statement A is incorrect. Okay, nanomolar to micromolar concentrations, we can use it. Then second is de novo, B's de novo sequencing is not possible by mass spectral method. Mass spectrometry is a technique which we use to determine mass to charge ratio of the ions. And basically, we can use it to determine the composition of our sample by measuring the mass to charge ratio of the ions, peptide ions. Now, what is de novo sequencing? De novo sequencing means it's a process of determining the sequence of an organism from scratch. Okay, from scratch. Without, so scratch means like you don't have a reference genome for it, without relying on the pre-existing reference genome. So that is de novo sequencing. So yes, we can use it. So this statement is incorrect. De novo sequencing is possible by mass spectrometry method. And here it is given not possible. So this is incorrect. Now, when I say B is incorrect, so whichever option having B or A cannot be the answer, right? So first cannot be the answer because it contains A, comprises of A and B. Then second also cannot be the answer, it has A. Then third has a B. So this is not the answer. So only you are left with option 4. Right? So this way you can solve this question. Let's check third and fourth statement. So C is the position of hydrogen atom in protein is not directly determined. This is correct. X-ray diffraction can provide you about electron density map, but it will not directly determine hydrogen atom positions because of low electron density. Hydrogen atom has one electron only, right? And D, circular dichroism and NMR resonance spectroscopy do not give the same information. This is also right. Circular dichroism gives you information about secondary structure and NMR is mainly used for tertiary. So statement D is correct. Okay, so if you don't know, please note it down. Circular dichroism is mainly, like if both are given precisely, circular dichroism is for secondary structure, that's alpha helix, beta sheet. NMR will provide you detailed information about three-dimensional structure information, including interactions, dynamics, whether your protein is in polar environment or non-polar, right? Protein, like its interaction, right? That can be studied with other atoms. So answer here is option 4, not 1, not 3. So Rinku and Sarita, is it clear to you? My answer is option 4, C and D statement is correct. Got it? Okay. Thank you for confirming. Now, this is our second last question. A researcher is investigating structural changes in a protein by following tryptophan fluorescence and by circular dichroism. Now, here it's a comparison which has been given. Like it's a here the two spectroscopy information, or you can say an experiment conducted using two spectroscopy technique. So this is a question which involves two spectroscopy technique. One is fluorescence and other is circular dichroism. Fluorescence is an emission spectroscopy technique. Circular dichroism is an absorption spectroscopy technique. Now, it is mentioned that fluorescence and circular dichroism spectra of a pure protein were obtained in absence of any treatment. Okay, that is A, in absence of any treatment. B is in presence of 0.5 molar urea. C is upon adding acrylamide, which is a quencher of tryptophan. And D is upon heating. And data for both are given below. Okay, so this is for fluorescence, this is for circular dichroism. Now, which one of the following statement is correct? See, these terms are very important: correct or incorrect, true or not true, right? Accordingly, you have to answer the question. So go through each statement and let me know what do you think is the correct answer here. So if you see, first and second are contradictory. Circular dichroism more sensitive to structural change than fluorescence, or fluorescence is more sensitive to structural change than circular dichroism. This is contradictory. Or both are equally responsive. Or fourth, acrylamide alters the secondary structure of the protein. Okay, option 2 you are saying, Sarita? Fine. What about you, Rinku? You don't have to think out of the box. Simply see the result given. So in fluorescence, you can see this is how we are getting the spectra when in absence of any treatment, then in presence of 0.5 molar urea like this, in presence of acrylamide like this, and on heating a straight line. And if you see circular dichroism spectra, like they are more or less same only, not much change, right? For in all three conditions, and D is on heating. So on heating, obviously the protein denatures. Biomolecules are very much susceptible to environment change, right? Change in pH, change in temperature, addition of denaturant, detergents, detergent concentration, temperature. So, Rinku, you are saying option 3. So, first of all, if you see both the spectra, definitely both are not equally responsive, because you can see here A, B, and C are not in under these in these three conditions. You are not seeing considerable change here in circular dichroism spectra, right? So obviously answer cannot be option 3, because if you compare both the graphs, they stand for different treatments and changes in structure of protein. So ellipticity graph is similar in all condition A, B, C, but if you see here, there is a considerable change, right? Different graphs are there for this is A, this is for A, this is for B condition, this is for C, right? So definitely fluorescence is more sensitive to circular dichroism. So I told you 1 and 2 are contradictory. So answer is option 2. Why? Because it is definitely we can see like different graphs on different treatments, which is not seen in case of ellipticity graph of circular dichroism. So answer here is option 2, and both are equally responsive. This is again incorrect. And again, like we can, like based on the data given, based on the result given, answer 2 is most appropriate. Then this is the last question. The figure shows fluorescence emission spectra of 3 different protein X, protein Y, and protein Z, excited at 280 nm. Which of the following statements gives the correct interpretation? First, protein Y and Z have tryptophan, while protein X only has phenylalanine. Second, protein X has only tyrosine and protein Y has tryptophan on the surface, while protein Z has tryptophan buried inside. Now, see, it's a fluorescence emission spectra, right? So, a few minutes back, we have discussed one question where fluorescence emission spectra was given, and I told you that how the tryptophan, if it is exposed on surface, right, from tertiary to secondary structure, like when SDS treatment is done, anionic detergent is added, then how it affects and results into change in spectra. So keep that point in mind. Third is protein X has tryptophan buried inside, while protein Y and Z have tryptophan on the surface. And fourth is protein X has only tyrosine and Y has tryptophan buried, and protein Z has tryptophan on the surface. So quickly give me the answer for this question. You just have to recall about what does the tryptophan, when buried inside and when exposed on surface, affects the absorbance, like affects the emission, like wavelength. With context to wavelength, you have to remember. Okay, you are saying option 4, Sarita. Rinku, you are also saying option 4. Fine, let's check. So only one thing that you have to remember is, as I told you, with respect to tryptophan, with context to tryptophan, only you have to remember. See, first of all, the absorption of light in protein is mainly occurred due to aromatic amino acids, tryptophan, tyrosine, and phenylalanine. And talking about tryptophan, so tryptophan fluorescence spectra, when we talk about, so always remember, tryptophan fluorescence lambda max information is used to assign tryptophan whether it is buried inside or it is exposed. So if the tryptophan is in a non-polar environment, okay, so as buried and in a non-polar environment, lambda max is less than 330 nm. But if it is longer than 330 nm, the tryptophan can be assigned to polar environment. That is, it is intended to imply solvent exposure. Now, if you see the X, Y, and Z, X is in this range, right? Like 300, starting from 300. Y is like 3. So you need to see near to 330 nm. So if you see the Y is like 320, like you can say it's 330, right? And here it is, this Z is beyond 330, right? So based on the information given, you can say that the Y indicates that the protein has tryptophan buried and the Z protein has tryptophan on the surface. So just based on this information, you can answer this question. And therefore, answer is here option 4. So Sarita and Rinku, you have answered it correctly. So well done, those who have solved the question during the live session. And I hope you have got some confidence. Many questions have been answered correctly. So Sarita and Rinku, especially, I hope you can analyze your preparation and has have got some confidence getting by getting answering the questions correctly. And everybody, those who have attended this session, throughout 1 hour session, you will be getting a freebie, a practice questions as a freebie. So the link of which will be shared in the chat. So it is already shared. Okay, you can go through that and practice more questions. So not only questions, explanation and the solution, like detailed explanation and the answer is being also mentioned along with the question. So keep studying. All the very best and take care. So with this, I end this session now. Thank you.